So today I will talk you about how we boosted multiphoton 3D printing technology for biomedical engineering. And our main focus was to create small features, but to give them high impact in the biomedical field. In this talk, actually, I would like to invite you to take a journey with me into the world of small features, features smaller than even the diameter of a hair. Today, I will show you how we made such small features using lasers and how we boosted these features to give them a high impact in bioengineering applications. As Catalina was saying, I'm working here in the National Institute for Laser Plasma and Radiation Physics and Center for Advanced Laser Technologies department, which is in this institute, and our institute is here very close to Bucharest, capital of Romania, in a small city called Magurele. So you can find us here. Now, to begin my talk, we thought that it's a good example to begin our discussion today. We took the concept of domino effect. It is not just a game, but it also has a scientific background, because you can see here scientific papers has been published on this effect. And this paper says that if you take a domino piece of 5 centimeters tall, it can topple another domino piece which is 1.5 times higher in volume. Based on this principle, if we add more and more domino pieces, one after the other, like here, the domino piece number 23 would topple the effect tower. So, small features, high impact. Our purpose to have such an approach was to use small structures, nano-structures, fabricated by a standard and well-known laser technique known as laser direct writing via two-photon polymerization, and to advance some new approaches with good results in especially in tissue engineering and drug delivery systems that I will show you later today. Our strategy relies on the fusion between reverse engineering and laser processing. What we did actually is, because nature is the best optimizer of the world, we took examples from nature, from the natural structures encountered in natural environments. We imported them in our standard 3D printing laser technology. The fusion between these two emerged in some very interesting micro and nano structures that resolved some of the current challenges in medical treatment. To reach this goal, we use an equipment which is called nanoscribe. It's a kind of 3D laser lithography system using a femtosecond laser with a high repetition rate, and which is focused on the sample through an inverted microscope, through different microscope objectives as a function of the spatial resolution that we want to achieve, as I will explain you in the next slide. This system is available at CETAL facility, and it's working continuously since the last 10 years, and it's still working very well. This technique is known for several advantages, such as we can get 3D structures with spatial resolution, lateral resolution below 100 nanometers. We can process materials below the diffraction limit, and which is very important for biological studies, for in vitro studies, is that our structures are fully reproducible because they are assisted by CAD. The physical principle behind the 3D printer I showed you before is two-photon polymerization using laser direct writing. Briefly, we put a drop of photoresist on a glass substrate, or it can be other type of substrate, not only glass. And on this photoresist, we focus very tightly a femtosecond laser beam. Here, at the focus point of the laser, polymerization takes place. This is the only place that the polymerization occurs, within the whole volume of the photoresist. And this small volume, which is polymerized, is called voxel. Voxel is actually known as the elementary unit of any kind of 3D structures printed in this way. Here is an example of our voxels for one of our 3D structures that we made. For instance, here you see a lateral size of the voxel of only about 300 nanometers, but we can also go below that dimension. Here you can see on the right a 3D microstructure printed using this technique, and above you see the simulation of how this structure is printed voxel by voxel. Well, using this technique, we have and started from, let's say, the quite well-known 3D micro nanostructure that have been extensively developed over the last few years in the field. We decided to boost these structures and to give them additional properties in order to achieve some interesting topics in tissue engineering, drug delivery, and so on. I will start and I will go with you through each of these proof of the concepts. Actually, these are proof of the concepts that we demonstrated. The first one is our first inspiration came from nature. That came from nature was that coming from the fact that we all know that small children and animal babies likes to move. Why? Because nature designed them in such a way, because nature knows that to grow and to strengthen the skeletal system, the bones needs to move. This principle was already downscaled to several cells, bone-forming cells, which are known as osteoblast cells, and it was found actually that if we move the cellular nucleus and we induce some changes here in the shape of the cell nuclei, we can improve the osteogenic effect, so we can fasten the bone-forming processes. We address this issue by a synergy between 3D topographies that we made by laser lithography technique that I showed you before. And we seeded cells on top of these topographies, and we just moved them in an ultrasound bath. This is the base principle of this part of our work. In a starting condition, we have made vertical microtubes of different heights, and we placed the osteoblast cells on them. On starting conditions, normally the cell nucleus have an almost spherical shape. In contrast, on the highest microtubes that are also ultrasonicated, so they are submitted to mechanical motion, you see here a strong deformation of the cell nuclei. This is also what we monitor in our experiment. These are the scanning electron micrographs of the 3D structures that we made, the microtubes with different heights. And here is an in vitro test with osteoblast-like cells seeded on these microtubes. You can see already here that we have a different behavior as a function of microtube's height. As the microtube's height increases, we have here much stronger deformations because the osteoblasts are acting with some traction forces here. You can see on the simulation what happens actually, as I was saying, to the osteogenic cells pull the substrate through these kinds of traction forces that they exert. And they induce strong deformation of the cell nuclei, which further induce strong deformations here, as according to the microtube's height. These are the cells which saw which are seen in fluorescent images, the entire cellular body with phalloidin and a cell nucleus with host fluorescent markers in static conditions versus ultrasound stimulation. And here we extracted from the image from the left only the cellular nuclei to see better what's happened with them. What we saw experimentally, that stronger cell nuclei deformation takes place on the microtubes which have the highest height, and plus which are under ultrasound stimulation. We quantify this effect. Look here first. We compare the shape of a nucleus on a flat substrate versus a nucleus before, as I told you earlier, on a topological surface. We had two parameters that enabled us to measure, quantify this shape of the cellular nuclei. And the conclusion was that, you can see here, the highest microtubes that were also subjected to mechanical stimulation underwent the stronger nucleus deformation effects. We checked if this effect of changing the shape of the cell nuclei are positive for our final goal, which was to improve the osteogenesis. For this, we checked two osteogenic markers, alkaline phosphatase and osteocalcin. Both of them showed that the best result was obtained here in yellow marks. For the highest microtubes, they were subjected to mechanical stimulation that I recall you, corresponding to the highest deformation of the cellular nuclei. Well, we went a little bit further. We had another approach. As you can see here on the right side of the image, we have a 3D structure that holds a weight of more than 10 kilos. The funny thing here is that this structure is actually made by spaghetti. We would not expect that spaghetti by themselves to be able to hold such a weight. The secret here is the hierarchy, the architecture and the hierarchy of this architecture. So if we take spaghetti and we arrange them in a very calculated and precise way, they are able to support 3 kilos. The same way happens with our structures and our cells. Using this 3D lithography technique that I showed you at the beginning of my presentation, we fabricated microstructures having such an architecture that they are able to support the growth, the attachment and growth of osteoblast cells that are very strong attaching cells, and they, as I showed you earlier on the slides, pull with a very high forces on the substrate. So our structure this time, we want it to be very strong from the mechanical point of view, so they support the cell growth. Another issue that we wanted to check, if we want to guide the cell adhesion at a specific place inside the structure, especially inside the structure, because usually when we're talking about scaffolds or implants, the problem is that the cells generally attach on the outer side or on the outer parts of the structure, and inside they do not attach, or if they attach, they do not interconnect. And as such, here inside, this generally form a necrotic core, which finally leads to implant failure. To avoid this, we tested several types of microstructures. For example, we have here some electron micrographs of our structures in a micrometric scale for the moment. We have chosen some two shapes, like ellipsoidal here and hexagonal here, to be as close as possible with the shape of a normal osteoblast-like cell, which is a polygonal shape. We made in vitro tests on the same osteoblast cells like the previous studies. We have a first bad result, which actually we expected it. The cells mostly attach on the outer side of the structure, and they were struggling to get inside, but with less success because there is no room inside the structure because the layers are too close to one another. We have another conclusion here, maybe not so useful at this point, but maybe later, because the cells, as you can see here, like more round edges than the sharp ones. We went further and we tried to improve our structures, so to give the cells a chance to enter inside. For this, we just made a simple thing. We added some more space between the horizontal layers of the structures. And what happens, you can see here, it's already much better because the cells appears to enter inside the structure in the free spaces in it. What happens actually here, what changes is porosity. The biologists know, and not only them, that bone is a spongy-like, has a generally spongy-like structure. So the bone cells need quite high porous structures to develop on them. As such, in the following study, we managed to fabricate the same technique, printing technique, a honeycomb-like structure made of some vertical microtubes and separating some layers with different distances between the layers. So what we managed to do here is to control fully the structure's porosity. And we want to see what porosity is the best to host osteoblast-like cells and what porosity is the best to support these cells to form bone tissue. Based on the simulation of the structure, we were able to calculate the porosity for each of these arrangements, partial arrangements of the layers. And we cultivated cells on each of the previous structures. We saw that the worst case scenario is here, where we have no spaces between the layers. The cells go inside each round hexagonal shape here, but they are separated. They do not interconnect, and as a consequence, they die. The best case scenario is here, where the cells attach not only on the surface, but also they appear to form some networks inside the structure. Taking the best case scenario from here, this porosity, we monitored a little bit what happened with the cells from the early stage of cell sitting on the substrates towards later stages. You can see how they develop. They are here, they still divide, they start to attach, and then they start to penetrate inside the structures. The same as before, we made the same measurements on these osteogenic markers, and we saw indeed that the best structures in what concerns porosity are the ones with porosity more than 90% that allow, let's say, the fastest bone formation process. Another application that we took, so another proof of concept that we demonstrated, regards multi-scale architecture, because it was found in nature as well that the structures that have different elements of different length scales that overlap between them are very efficient for preventing the settlement of microorganisms. For example, they are very efficient as cell-repellent surfaces. The existing technologies that we have so now were quite complicated because generally they require multi-step procedures to have both micrometer scales and nanometer scales. In our case, the advantage of our 3D printing technology is that it's a single-step procedure that enable us to provide micro scale structures, micro scale structures in the end. Right in the same processing step to get some nano features on top. Here are two examples of microstructures in the shape of mushrooms, because we want to make them a little bit also water repellent. This was the reason. That are decorated with fingerprint-like structures. On the top part, we have microstructures here on the fingerprint, here nanostructures, which you will see better here in the atomic force microscopy image. Shows you better the nanostructures on top of this mushroom, which is easily given by the fact that our voxel has a spiral-like fashion inside the photoresist to polymerize it. And here you have the microstructure compared micro with nano numerically here from atomic force microscopy images. We cultivated cells on these two substrates, microstructures and micro plus nanostructure. So these are the hierarchical structure. Both of them were actually good in repelling cells, as you can find it here. But the best were still the nanostructure substrate. So we have a microtopography decorated with nanotopography on top. So we have a hierarchy topography here. We show the best results in what concern their capacity to repel cells. I will quickly summarize our next approach. Using this idea of hierarchical structures, we looked again to examples from nature, and we found that nocturnal insects such as Grapholina funebrana moth, they have the eyes like this. They have some ommatidia-like structures, which are hemispherical ones in micrometer range, which are covered with some nanopillars. These nanopillars have the role of decreasing the reflectivity in the infrared, so during the night, because these insects do not want to be detected by predators, and also to increase the transmission in the infrared, because these insects have the need to have a good vision in the infrared, so during the night. We took this example and we replicate almost the same structures using our 3D printing technique. We obtained these structures. Here are the natural ones, and here are the ones that we obtained by laser. And we tested their optical properties, a little bit the focusing. I will not stay too much here. And also, as I told you in the beginning of this idea, the reflectivity decreased and the transmission increased. So the functionality in the infrared, so the functionality of our structures was validated. After this, keeping in mind all the micro nanotopographies, as you saw up to now, our next thought was to improve a little bit the interaction between the structures and biological materials, in our case, the cells. To do this, we try to coat our structures with some materials which are of course biological compatible, and also they are very good as osteointegration, osteogenesis processes. The problem is here that the two materials that are very good for bone regeneration are very difficult to process. So it would be quite impossible to process them directly into such 3D structures. Instead, we made the backbone of the structure by commercial photoresist biocompatible one, as we did before, and we coated these structures by immersion in collagen chitosan blends. And we saw what happens with the cells that were seeded on this coated structure. I will not stay too much on this part because there is much room to improve here regarding what happens with cell attachment, but I will tell you that we tested different blending ratio between collagen and chitosan. The same test with the osteogenic markers as before, the two ones here. And we have found that the best results from this point of view of bone regeneration, or at least bone formation, was obtained for a collagen chitosan blending ratio of 20% to 80%. Then we look to another direction to see a little bit what happened with bone implants in general. These are affected in many cases by chronic inflammation after implantation, of course. And this is treated by administering anti-inflammatory drugs such as dexamethasone. But dexamethasone is generally administered by oral routes, which are toxic for the body and have severe side effects. We tried to respond to this difficulty by fabricating very small micro reservoirs to be located next to the implant, and also to load these micro reservoirs with dexamethasone and to release the dexamethasone on demand and specific time scales by applying external electrical stimuli on these structures. As such, these are the main steps we took here. We first fabricated by laser printing the microreservoirs. We filled them with polypyrrole, which is an electrical conductive polymer, and dexamethasone. We sealed them with PLJ, which is a biodegradable polymer. We did this to prevent the passive diffusion of the drug. Then we placed this between electrodes. We seeded osteoblast-like cells on these structures, and then we tried to release the drug by externally applied electrical stimuli. These are our structures seen in scanning electron micrographs, and their dimension then along with their volume. The volume is important related to the amount of drug that it can be loaded into each of these microreservoirs. You can see here a nice top view image with the microreservoirs empty, then partially filled with polypyrrole and dexamethasone, and then coated with PLJ layer. The most important result here is that in gray, it's a passive drug release, and in color, you can see a step-like fashion behavior of the drug release that takes place in a step-like way, according to the green arrows that correspond to the applied electrical stimuli on these samples. Again, we tested the final goal actually of this trial is to see what happens with the two osteogenic markers, if they are increased or not. We saw that for the electrically stimulated samples here, you have a higher LLP activity and higher osteocalcin activity for the electrical stimulated samples as compared to the non-stimulated ones. Finally, but not the least important, you can see here another proof of concept which involves magnetic fields. The image here is, of course, magnetic levitation, which, okay, will not be the case in for our cells, but we use the fact from nature that we all live in magnetic fields, actually. And it was actually found that magnetic fields can interfere with even cells if the magnets around the cells are small enough, so the gradient fields around the cells reach the same size as a cell. For example, let's say around tens of micrometers. And there is a difficulty in our days to scale down such magnets to make them as small as a cell. We took advantage of our 3D printing technique that allowed us, as I recall you from the first slides, we can get very small switchers with very reproducible architectures and nanometer and micrometer scale. But because it seemed to be quite complicated to make them magnetically active, we started with 2 dimensions. First, because 2 dimensions are also important, for example, for studying individual cells for long time scales or to study the communication between cells and so on. In brief, what we did, we used the 3D printing technique that I told you in the beginning of my presentation. Then we did a chessboard-like structure where you have here the gray squares with just photopolymer, non-magnetic, so which was alternating with the brown squares that were magnetic, because here was photopolymer blended with super paramagnetic nanoparticles. If we seeded fibroblast cells on this chessboard-like structure, without any kind of magnetic field around, the cells attach randomly all over the place. On the opposite, when we place the chessboard-like structure in the static magnetic field, the cells preferentially attach on the magnetic squares. This is the design of the experiment. This is actually what we did. These are semi-micrographs of our structures, magnetic, non-magnetic. Together, they make the chessboard-like structure. And we wanted, of course, to check if we have magnetic or these nanoparticles inside the structures at the position where we want them to be. We did this by enhanced dark field microscopy, and then we checked if we have magnetic nanoparticles or the presence of iron. We did this validation by EDAX. The in vitro tests showed the following conclusion. If we have no magnetic field applied, as I told you in the first slide explaining this experiment, the cells attach all over without any kind of preferential attachment. On the upper side, we applied static magnetic field. The cells mostly attach on the squared magnetical areas. The better view is here where you see only the cell nuclei marked with Hurst, because we have also autofluorescence of our photopolymer, which disturbs a little bit the whole image here, where you can see the cell in red. But the one with blue cells nuclei, it's very suggestive. A nice and view, but the same result can be seen here. It's without static magnetic field. It's a 3D view here, top view and lateral view. No preferential attachment of the cells occur, but if we apply a static magnetic field, you have this attachment of the cells only on the magnetical areas. Since we validated our proof of concept in 2 dimensions, then we were able to pass to 3 dimensions. We made 3D structures where we incorporated in the volume of the structures magnetic nanoparticles, the small dots here. When we cultured cells on these 3D structures and we applied external magnetic fields, we wanted to see what happened. A first observation, it was unexpected, but it was good. Without magnetic nanoparticles, the photopolymer we used here did not allowed us to have good structures. The structures collapsed. On the opposite, when we started to add magnetic nanoparticles inside the photopolymer, you see that with increasing magnetic nanoparticles concentration, the mechanical resilience of the structure improved. Again, as in the 2 dimensions, in 3 dimensions, we wanted to see if we have magnetic nanoparticles, or at least, okay, now we don't know yet that they are magnetic, that at least to see that we have some nanoparticles inside the volume of the structure. We use enhanced field microscopy images, and you can see here in red dots, of course, false colors, you have the nanoparticles within the polymer, which is marked with green. We checked again in this, also in this case, a 3-dimensional case, that we have magnetic nanoparticles. You have here EDAX measurements. And the in vitro test, briefly, I will just show you briefly. We have used here a dye, alizarin red staining, which shows zones of mineralization within the tissue in the shape of calcium phosphor, like in the natural bone, and the best result was here. So when we have a structure with 4 milligram per milliliter magnetic nanoparticles inside and exposed to static magnetic field. Now, I will summarize my talk saying that the small structures that we make by 3D lithography can have high impact in biomedical applications, such as in our case, tissue engineering or drug delivery, but we have to keep in mind that these small features also raise quite high challenges that we still need to overcome. As perspectives of our research, we want to upgrade our proof of concept to biodegradable polymers. We want to advance with these concepts towards in vivo experiments, and we also want to expand our research to other cell lines. I would say we are mostly interested in neuronal cells. Before I come to the end of my presentation, I would like to remind you that CETAL is a new laser facility part of Laser4U. We are more than happy to invite you to visit us and to start collaboration with my colleagues from all 3 laboratories that we have here in CETAL. You can find us here if you wish, or to contact Felix Sima, of course. In the end, I would like to express my gratitude to Felix Sima for giving me the honor to have this talk. And I would also like to thank to my good colleagues here, for their important contribution to the results I presented today. Especially thank you to Bogdan Călin.